public PhD defense of Elizabeth Perona Vico. Maybe I would ask everyone to mute the microphone when someone else is speaking, because I hear some background. Okay, thank you. I think it works. Oh. I just mute myself. I mute Elizabeth. I think that was making the echo, but I will give you the room very soon, Elizabeth. So welcome all to the thesis of Elizabeth Perona Vico. We'll have her public defense today. The title of the thesis is Importance of Hydrogen-Mediated Mechanisms for Microbial Electrosynthesis Regulation at Molecular Level. And before we go to the presentation, I will introduce the elements of the jury. First, we have Professor Hélène Vu. She's Professor of Electrochemical Engineering at the Chemical Engineering Department from Loughborough University. And then we have Dr. Annemiek ter Heijne. She is Associate Professor at the Department of Environmental Technology from Wageningen University and Research. Myself, I'm Diana Souza, and I'm Professor of Microbial Physiology at the Laboratory of Microbiology from Wageningen University and Research. And with this, I will now ask Elizabeth to take the word and to show us her work, her PhD work. Elizabeth, you have maximum 1 hour for this. So we are looking forward to your presentation. I'll give you the word. Try unmute. Elizabeth, you have to unmute yourself because I'm muted, but now I cannot unmute you.
Now? Okay, thank you so much. So, good morning. Thank you all for being here, and thanks to the committee for the introduction. This thesis defense will be divided in 7 different parts, starting with the introduction and objectives. I will follow with the main 3 chapters, each one composed by a background, experimental design, and results and discussion section. And to conclude, I will discuss altogether the presented results and read the conclusions. Microbial electrochemical technologies, from now on named as METs, are the focus of this work. They are defined as technologies or applications that utilize the electrochemical interaction of microbes and electrodes. METs are conducted in bioelectrochemical systems, or BS, consisting on 2 electrodes, a cathode and an anode, separated or not by an ionic exchange membrane. Usually, an aqueous solution is surrounding the electrodes, enabling the operation and bacterial growth. The first proposed application of BS was microbial fuel cells, where microorganisms present in the anode could produce electrical energy at expenses of waste or organic substances. In other systems, the energy provided by a microbial fuel cell reaction can be used for hydrogen production in microbial electrolysis cell, to drive desalination processes in a microbial desalination cell, or to synthesize organic compounds in microbial electrosynthesis. In this thesis, microbial electrosynthesis, or MES, was the main focus. As stated before, MES is engineered to use electric power and carbon dioxide as the sole energy and carbon sources in reductive bioelectrochemical processes for the biosynthesis of several compounds such as acetate and butyrate, but also some alcohols like ethanol. Both complex microbial populations and pure cultures have been used for this purpose. The most promising part of this technology is that it serves as carbon capture and utilization platform, where the products might be used to be converted in more complex substances such as long carbon chain biochemicals or biofuels that can be used in the industrial sector. Interestingly, MES and all the other microbial electrochemical technologies take advantage of the ability of some microorganisms that directly transfer electrons from a poised electrode. This group of microorganisms able to transport electrons in or out their cell walls and then able to interact with the near environment are named as electroactive bacteria, and they can perform extracellular electron transfer during anaerobic respiration. Depending on the ability to release or uptake electrons, bacteria can be classified as electrogenic or electrotrophic. In biocathodes, there are different mechanisms for extracellular electron transfer, being direct electron transfer through direct contact between microorganism and cathode or via conductive pili or nanowires, mediated electron transfer via endogenous or exogenous electron shuttles, such as flavins or phenazines, and indirect electron transfer via hydrogen or formate generated electrochemically, hydrogen enzymatically produced by extracellular hydrogenases, or hydrogen or formate produced by a syntrophic interaction with a microorganism able to direct uptake electrons from a poised cathode. Since hydrogen is the cornerstone in indirect electron transfer, we focus this thesis on studying at what extent hydrogen is a limiting factor or an opportunity to new approaches in microbial electrosynthesis, mainly focusing our efforts on understanding better the microbes participating in this technology. Knowing this, the specific objectives were to identify archaeal and bacterial enzymes potentially involved in direct electron transfer in enriched biocathodes, to analyze the electrochemical behavior of several model bacterial strains and their ability to improve hydrogen evolution in biocathodes, and to define a strategy towards genetically modified supervibrio to achieve higher hydrogen titers in BES. Each one of these objectives is linked to one of the chapters in this thesis. In the first chapter, we studied the effect of transient changes in electron flow at the expression levels of some genes putatively participating in extracellular electron transfer in an enriched methanogenic community. The term electromethanogenesis was first defined as the reduction of carbon dioxide to methane using an electrode as the electron donor. After several years, hydrogen-mediated methanogenesis was included in the definition, since more and more studies pointed out the participation of hydrogen as the main electron transfer molecule. Therefore, if we get back to the known extracellular electron transfer pathways and we illustrate it for methanogens, both direct and indirect electron transfer could be taking place during electromethanogenesis. Indirect electron transfer events could occur via abiotic or biotically produced hydrogen and also by other compounds such as acetate. If we consider the possibility of direct electron transfer in methanogens, several outer membrane redox proteins have been pointed out as being responsible for electron uptake. These enzymes are cytochrome C-type in the RNF complex, as proposed for the acetotrophic methanogen methanosalcina aciduvorans, or membrane-bound and cytoplasmic hydrogenases, and also formate dehydrogenase, as proposed for Methanococcus maripalolis. In this study, we tried to understand better which mechanisms were involved in the communication between the electrode and the cells during electromethanogenesis. For that, we studied how short-time exposure of open electric circuits affect to the expression levels of genes coding for several enzymatic complexes, being mainly hydrogenases. Experimentally, we had a 3-neck bottle BES with a nominal capacity of 1 liter with 4 cathodes connected independently. Cathodes were made of carbon cloth. The effluent of another reactor operated at similar conditions was used as inoculum for this experiment. Operation was performed at -1 volt for 53 days. During this time, electrochemical characterization was performed measuring both the gas and liquid composition and cyclic voltammetries were performed in abiotic and biotic conditions. The configuration of this BS allowed us to maintain 2 of the electrodes disconnected from the current for 6 hours at the end of the operation, enabling the analysis of the expression levels of the selected genes. After that, samples of biofilm and bulk were taken and nucleic acid extracted. DNA and RNA were used as proxies for the total and active microbial community. Microbial composition was analyzed by Illumina-based 16S sequencing. Primers were designed specifically for each gene, and RT-PCR was used to determine the abundance of these genes between open and closed electric circuits. For this study, we had the objective to set up a simple system able to sustain methane production by electrochemical. As shown here, methane remained stable during the experiment in a volumetric concentration from 70 to 90%. Hydrogen was produced during the first days of operation, and after that was completely depleted, mainly because it was consumed to produce methane. And acetate otherwise was produced between days 7 and 10, and after its presence was minimal. Electrochemical characterization was performed using cyclic voltammetries that consisted on analyzing changes in the slope of current demand versus cathode potential. Cyclic voltammetries were performed before inoculation in abiotic conditions, shown here in gray, and in biotic conditions, marked here in black, for the 4 biocathodes independently. Further analysis was performed to estimate cathode potentials at which redox reactions were taking place. In 3 of the 4 cathodes, a redox pair was clearly identified around minus 0.7 volts, which is within the range of redox potential values generally assigned to the activity of hydrogenases. Going to the microbial community composition, according to DNA analysis, the used inoculum was composed mainly by the orders Methanobacteriales, Burkholdeirales, and Rhizobiales. But in contrast, biofilm attached to the cathode resulted in a highly enriched population of methanogenic archaea, where Methanobacteriales remained between 70 to more than 80% of the microbial population, and also was predominant in the bulk liquid but remained at lower proportion. Therefore, higher enrichment of methanogenic archaea was detected in the BES, especially in the biofilm. The genus level, all members of this order belonged to the genus Methanobacterium. Complementary DNA, or cDNA, synthesized from RNA, was used as proxy to identify the active members in the biofilm community under open and closed electric circuits. Here is shown a principal coordinates analysis plot representing each dot as the community structure for each sample type. Considering the composition of the samples in the disposition, of the samples in the plot, the active microbial community of connected samples seemed to be different from the disconnected one. But it is true that only 2 samples for each condition could be tested. If we look at the relative abundances of the microorganisms, Methanobacterium-related sequences appeared at lower densities in cathodes that remained disconnected than in the cathodes that were connected. In this sense, we perform a statistical analysis with the objective to elucidate if any of the taxonomical groups was present in a different relative abundance between the 2 conditions, and no statistically significant differences could be confirmed. Overall results presented since now demonstrated that Methanobacterium was the main responsible archaeon conducting electromethanogenesis in our system remaining in active state. Therefore, was used as model organism. As explained before, one subunit of each enzymatic complex was selected to compare its expression level at off and on or open and closed electric circuits. Here, the plot shows the relative abundance between the selected subunit respect one housekeeping gene. In the upper plot, it is shown the proportion using the 69 gene, and in the lower one, the cell division protein FTSD. Empty bars represent open circuits and filled bars closed electric circuits. Starting with the membrane-bound nickel iron hydrogenases, EHA and EHB, expression levels were higher when electron flow was interrupted. Slight differences between the two conditions were found in the analyzed subunits for cytoplasmic hydrogenases when normalizing the results with the 16S RNA gene, but between 1 to 1.5 increase in open circuit conditions was detected when FTSD gene was used. Similar results were found for the expression of cytoplasmic expression formation protein. Increased relative abundances for almost all the genes were obtained under off conditions, but no statistically significant differences were found between the two experimental conditions. No results are shown for 4-methanofuran dehydrogenase and formate dehydrogenase, since no specific amplification could be obtained with the design set of primers. So, overall results demonstrated reduced changes in the relative expression of the selected genes, and at the time that we published this chapter, we highlighted different aspects that could be explaining it. First, since two of the electrodes remain connected to the current in the same cathodic chamber, enough hydrogen could be produced, maintaining minimal levels of available reducing equivalents, preventing significant changes in the gene expression. In our case, no further tests were performed, but the presented results, even not being significant, suggested the opposite idea, because hydrogenases tend to be overexpressed under hydrogen deprivation. Second, the used 6-hour exposure to the open electric circuit was not enough to trigger significant changes in expression levels, suggesting that longer exposure times to on-off electron flow might be tested to confirm changes in the relative expression of hydrogenases. And third, the test enzymes were not participating in electron capture in methanobacterium, or they were not sensitive to changes in electron availability. Then it's clear that direct and indirect electron transfer events may happen simultaneously, but methanobacterium seemed to support its activity on the utilization of hydrogen. Several authors have been pointing out that bioelectrochemical processes during MES are enhanced by a proper hydrogen availability in the cathode compartment to avoid one of the main limitations in MES, that is the rate at which microorganisms acquire electrons. So we thought about hydrogen as an opportunity for new experimental approaches and not as a limitation. In this second chapter, we experimentally tested the ability to biologically produce hydrogen of several pure cultures to further improve microbial electrosynthesis. In biocathodes, hydrogen can be produced either abiotically, where the reaction is thermodynamically favored at minus 0.6 volts in carbon-based materials, or biotically, with the participation of living microorganisms of both non-defined, mixed cultures and monocultures, or isolated enzymes. Some studies have proven that hydrogen is biotically produced for several species, such as Geobacter sulfurreducens, Rhodobacter capsulatus, the sulfurvibrio species, among other protobacteria. Also, increased hydrogen productions have been detected in the presence of cell-exhausted medium from cultures of Sporomus aubata, Sporomus asperioides, and Methanococcus maripaloides. The former presence of microorganisms in the cathodic chamber led to the electron modification via deposition of metals or free enzymes, such as hydrogenases and formate dehydrogenases released by the microorganism. This part of the thesis has been developed in the frame of the European project BioRecover, in which one of the main objectives was the selection of suitable candidates to establish resilient co-cultures, ideally composed by a hydrogen-producing strain in combination with an homocetogen for microbial electrosynthesis processes or electrofermentation. In this case, co-cultures were thought to be performed with genetically modified strain property of one of the partners of biorecover global bioenergies. Part of the time during this thesis has been used to test these strains, but no results could be shown since they remain under confidentiality. In any case, the first step of this project was to achieve a resilient hydrogen production platform. So we aim at assessing the capacity to produce hydrogen in biocathodes using 10 strains from the genera Rhodobacter, Rhodopseudomonas, Rhodothyclus, Sophovibrio, and Sporomusa. Following with experimental design, bacterial strains used can be grouped as purple non-sulfur bacterium, an homocetogenic, and sulfate-reducing bacteria. In this case, BS was an H-type cell with a nominal capacity of 150 milliliters. A sole electrode made of carbon cloth was placed in the cathodic chamber, and anode and cathode were separated by a cationic exchange membrane. A hydrogen microsensor was placed directly in contact with the catholyte to perform online hydrogen measurements. A comparable methodology was used to treat the same way all the tested bacteria. This includes abiotic control, bacterial growth, and biotic BS operation. To operate the systems in both conditions, abiotic and biotic, we set the cathode potentials at -1, -0.8, or -0.6 volts. Cyclic voltammetries were performed at the end of these two steps. After abiotic controls, cathodes were incubated with bacterial cultures for 10 days to ensure biofilm formation in the electrode surface. Each of the bacterial groups were incubated in the most favorable conditions using a sole organic source. To conduct biotic BS, minimum changes on medium requirements to sustain bacterial growth were made, and carbon dioxide was used as carbon source. To elucidate if biofilm was formed into the cathode surface, pieces of electrodes were taken at the beginning and at the end of the biotic operation to perform DNA extraction and quantify the 16S rRNA gene by quantitative PCR. Abundance of this gene was used as proxy for bacterial density. Starting with obtained results for the cell densities in the biofilm, it was confirmed that all the tested bacteria were present since the beginning of the experiment, and the number of cells attached to the electrode increased over BS operation, except for Rhodopseudomonas and the market isolate. Even though, altogether, these results confirm not only the biofilm formation, but also its stability into the cathode surface. To evaluate hydrogen productions and ensure proper interpretation, production rates were calculated at biotic and abiotic conditions. For the biotic conditions, the productions were calculated after the first and the second carbon dioxide feedings that are shown here in yellow and purple, respectively. As we can see, the obtained results for purple non-sulfur bacteria had the same tendency after the first carbon dioxide feeding, where an increase on production rate compared to the abiotic were observed. But in the case of Rhodobacter species and Rhodothyclus, after the second feeding, rates remained slightly lower than in abiotic conditions. Among all the analyzed electrochemical parameters, here Coulombic efficiencies to produce hydrogen are shown. For the two Rhodobacter species, Coulombic efficiencies remain similar between abiotic conditions and the first feeding in presence of bacteria. But when testing Rhodothyclus, Coulombic efficiency decreased in the first feeding to remain like the abiotic after the second one. So hydrogen production was not sustained in time, remaining similar to abiotic conditions, possibly indicating indicating that hydrogen might be produced as a residual activity from photofermentative conditions used during bacterial growth in the cathode electrodes. But this is only possible if any intracellular reservoir was accumulated by these strains and mobilized under non-optimal conditions. In contrast, for reduced Pseudomonas and the two isolates, from 2 to 7-fold reduction from maximum production rates were found after the second feeding, and reduction on Coulombic efficiency occurred accordingly. Therefore, hydrogen production was not sustained in the time for these strains. The drastic decreased production rates with Rhodopseudomonas palustris and this isolate could be explained due to the already shown lower cell densities measured with quantitative PCR. So probably a major hydrogen consumption was taking place with these strains after complete adaptation of the cells in the BES. Thirdly, when Sporomusa ovata was used, hydrogen production rates decreased over the operation time, being 4-fold lower in the first feeding, and after the second, no hydrogen was detected. Sporomusa species were described as homocetogens, being able to produce acetate at expense of carbon dioxide and hydrogen. Remarkably, increasing acetate concentrations were measured during the experiment, achieving a final concentration of 6.4 millimolar. Therefore, the role of Sporomusa in the system was clear. It was consuming the produced hydrogen, possibly abiotic, and carbon dioxide to form acetate. Following with the obtained results with sulfate-reducing bacteria, production rate was significantly higher after the first feeding with the sulfovibrio sulfuricans, but slightly decreased where rates were obtained with the other two desulfovibrio strains. After the second feeding, production rate with the sulfovibrio desulfuricans decreased in contrast with the first feeding, but remained significantly higher in comparison with abiotic conditions. Coulombic efficiencies were lower than in abiotic conditions, but similar between the feedings. In the case of the sulfovibrio vulgaris, no conclusive results could be obtained since production rates remain similar than in abiotic conditions. Maybe extended operational times could elucidate if they are suitable as hydrogen-evolving candidates in biocathodes. The most promising results were obtained with the sulfovibrio pacesi, with a statistically significant increase of 8-fold in the production rate after the second feeding, and Coulombic efficiency remained between 80 to 90%. These results were similar to the ones obtained before by Aulenta and coworkers. Regarding the obtained results with these strains, short tests at less reducing potentials were performed. Operating the BS at -0.8 and -0.6 volts. Only one additional measurement was performed after a feeding with carbon dioxide. Under these conditions, only the sulfovibrio pacesi demonstrated higher and better performance than the other strains. Hydrogen production was not detected either in abiotic or biotic conditions at -0.6 volts. Electrochemical characterization was performed using cyclic voltammetries. Plots shown the abiotic and biotic curves in gray and black respectively. In 8 of the strains, the current demand of biotic cyclic voltammetry differed from the abiotic. Also in these strains, the current demand started to increase at potentials around -0.4 to -0.6 volts. That can be seen here as the shift in the biotic curve respects the abiotic. Then the cyclic voltammetries indicated some electroactivity for almost all the strains, but unfortunately this could not be translated into direct hydrogen production in all of the strains. Further analysis was performed to estimate cathode potentials at which redox reactions were taking place. Only in these 6 strains, a redox pair was found at around -0.6 volts, which as said in chapter 1, has been typically observed in hydrogen-producing biocathodes and related to hydrogenase activity. The objective of this study was to ensure a proper microbial platform to enhance hydrogen production in biocathodes and to further develop synthetic consortia for microbial electrosynthesis. Altogether, the presented results demonstrated that at least 8 of the tested strains presented at some extent electroactivity. Hydrogen could be biotically increased in biocathodes, but only sustained in the time by some of the test strains. And the most promising candidates to continue our research were the sulfovibrio strains. At this point, we didn't know too much about direct electron transfer events, but we knew that some strains were able to increase hydrogen production in biocathodes. In this sense, the sulfovibrio strains were taken as candidates to further improve hydrogen production by means of genetic engineering. This chapter was possible thanks to the collaboration with a group of Professor Miriam Rosenbaum in the Biopilot Plant Group at Leibniz Institute for Natural Product Research and Infection Biology. As stated before, the sulfovibrio strains belong to the sulfate-reducing bacteria group. These are able to generate energy using sulfate as the terminal electron acceptor while oxidizing hydrogen or various organics, resulting in the production of sulfide. As shown here, some of the key enzymes in the metabolism of sulfate-reducing bacteria are hydrogenases and cytochrome C type. Interestingly, several sulfate-reducing bacteria have been described as able to directly uptake electrons from the poised electrode, and despite the lack of specific knowledge, it seems possible that hydrogenases and cytochromes would be involved in direct electron transfer. Regarding the obtained results in Chapter 2, where these bacteria were producing hydrogen more efficiently, and the fact that there is available information about the enzymatic machinery involved, this chapter had the aim to increase the hydrogen production capacity of the sulfovibrio pachyci via overexpression of genes putatively implicated in the process. Selection of genes, vector construction, and definition of a workflow to transform the sulfovibrio pachyci were the main focus. The experimental design was divided in 2 parts. First, for the construction of expression vectors, sequences of the genes of interest were retrieved from the available genomes of the sulfovibrio vulgaris used as model, and the sulfovibrio pachyci, and specific primers were designed. Genomic DNA extraction from both cultures was performed, and PCR was used to amplify the genes. For the vector construction, enzymatic digestion, ligation, and transformation in Ecology were done. And to select the appropriate construction, a screening of the obtained clones was done, and candidate plasmids were sequenced to check the results. In the second part, different transformation protocols based in electroporation were tested to find proper conditions to overexpress the genes of interest in the sulfovibrio. Previously to the construction of the expression vectors, available sequences of the plasmid described before as suitable for the sulfovibrio were examined in silico. Plasmid named as PMO 9075 was selected as the best choice since its stability in the sulfovibrio vulgaris and other the sulfovibrio strains has been confirmed. The enzymes presented in the background section involved in the main metabolic pathways of these strains, hydrogen oxidation and sulfate reduction, were considered as putatively interesting to be our candidates. Annotated genes for the model strain, the sulfovibrio vulgaris, were used to find homologs in the available genome of the sulfovibrio pachyci, comparing the protein sequences with protein blast. 3 from the 4 periplasmic hydrogenases in the vulgaris were found in the pachyci. Only 1 membrane-bound hydrogenase was found, and cytochrome C3 was also present. For the selection of one of the hydrogenases, periplasmic hydrogenases were considered more interesting since they are located near to the extracellular environment. Iron-only hydrogenase was finally selected since it was described as putatively being more active during hydrogen evolution in the Sulfobibio vulgaris under fermentative conditions. On the other hand, since cytochromes have been described for several bacterial strains as being participating in electron transfer, cytochrome C3 was selected as candidate. We designed two plasmids with each of the selected enzymes, some using the genes of the Sulfobibio vulgaris and the others with the Sulfobibio pacesi. To optimize the transformation protocol, we started working with the Sulfobibio pacesi and the empty plasmid. Several experimental conditions were tested where the main variables are marked here in numbers. And first tests were named as A and B and D and E and were based on different protocols. The only fixed parameter between them was the initial culture volume. All the experiments were performed with and without the presence of iron sulfide precipitate resulted from the strange metabolism. The idea to remove it was taken to avoid any interaction of this molecule during the protocols. Only tests A and B resulted in visible colonies after transformation, but they didn't grow again when transferred in new media. Two more tests, named as C and H, were performed, this time increasing the amount of initial culture volume from 9 to 50 milliliters. Test H resulted in no transformants, but colonies were detected in test C. Again, the colonies were transferred to new media, but this time utilizing different concentrations of antibiotic. Unfortunately, growth after picking colonies was detected only for the lowest amount of antibiotic used, and therefore plates were discarded for further analysis. Since our reference protocol was described to be useful with the Sulfobibio vulgaris, we decided to repeat test C with this strain. Also, additional tests F and G were done using one of the constructed vectors. These tests resulted in more promising results since growth under antibiotic selection was detected and maintained after colonies were picked and transferred into new media independently on the antibiotic concentration used. For that reason, further analysis was performed with some of these cultures. Plasmid rescue and colony PCR were used to check for the presence of plasmids in the Sulfobibio vulgaris. Unfortunately, neither plasmid nor positive PCR amplification was obtained from any of the cultures. Altogether, the results were not really hopeful, but at the time of the submission of the thesis, we dedicated a section to critically analyze all the data and highlight some aspects to be considered for further experiments. And since my scientific training didn't stop with the submission of the thesis, we continued working and tested several of the proposals. First, we changed from post-gate medium to the one described by Keller and co-workers being efficient on their experiments with the Sulfobibio vulgaris. This change mainly affected the presence of iron sulfide in the cultures that was significantly reduced and allowed us to proper measurements of optical density to control growth of the cultures. In this medium, we did antibiotics sensitivity studies with both strains, and results show no growth at any of the used antibiotic concentrations, being both bacteria sensitive to spectinomycin. Also, we did two new sets of transformations with the Sulfobibio vulgaris using lower amounts of DNA, but none of them worked. At this point, we could test different voltages to increase the chances of electroporation, but we prefer to use natural transformation as a possible mechanism for introducing foreign DNA, even though no transformants could be detected. Therefore, by now, our idea is that our main limitation might be the initial number of cells, and if we could enhance them by controlling the growth conditions and kinetics, it could be possible to succeed with any transformation protocol that we implement in the sulfovibrio strains. Considering all the work done in this thesis, it is clear that hydrogen was the main character, being the key element for carbon assimilation during autotrophic growth, and also reported as the main intermediate molecule in microbial electrosynthesis. In this sense, this thesis highlights the importance of understanding deeply the extracellular electron transfer mechanism in biocathodes, how to enhance hydrogen availability utilizing microbes as catalysts, and the application of synthetic biology to improve PS performance. In biocathodes, mostly conformed by non-defined mixed cultures, it is essential to fully characterize the microbial community composition for a better understanding of the resident bacteria and/or archaea. For that, molecular techniques such as massive parallel sequencing of 16S rRNA gene are a very common and used technique to have an overview of relative abundances of the main players in mass. Taxonomic composition and phylogenetic diversity could be studied using DNA extracts. Also, absolute quantification of the gene marker could be performed using quantitative PCR. Otherwise, RNA analysis provides information about the putatively active community. This was the approach that we followed in Chapter 1. Deeper analysis could be performed using metagenomics, where the genes from the community are sequenced and putatively metabolic and functional profiles might be obtained. On the other hand, metatranscriptomics can reveal active expression profiles of genes or metabolic pathways at a given time and conditions. These two molecular techniques are pointed out as the most promising to further understand how microbes communicate with electrode surfaces. In fact, some approaches have been published from now. Here, the work of Ragat and co-workers is shown. The authors used both techniques, metagenomics and metatranscriptomics, to analyze potential induced changes in the expression levels of several genes in enriched electromethanogenic community, where the most abundant archaea were methanobacterium. As depicted in this scheme, no significant differences at transcriptomic level were found between the different time exposures to a less reducing potential. These results, at some extent, confirm the main results exposed in the first chapter of this thesis, where hydrogenase activity analyzed using RT-PCR resulted not being regulated at transcriptional level in methanobacterium. During mass, compounds with 2 carbons are the most produced chemicals, while compounds with 3 to 6 carbons remain at small titers or are produced intermittently in the time. In this sense, hydrogen as the principal intermediate molecule to enhance mass processes is considered during this thesis. However, to what extent hydrogen is the main bottleneck is still to be elucidated. So in this thesis, we move from non-defined mixed cultures to starting working with monocultures as the first step to develop synthetic co-cultures as a potential solution to overcome the presented limitations. The use of defined co-cultures in mass is scarce, but one feasible approach was described before. Utilization of the sulfopylia corrodens as in situ hydrogen producer in combination with Methanococcus malipaludis or Acetobacterium goodi resulted in enhanced methane and acetate production respectively. Then the presented results in the second chapter could be considered a significant step forward to the further application of the sulfovibrio strains in synthetic co-cultures. Genetic engineering, understood as gene editing and control of gene expression levels in microorganisms, is attractive in the field of industrial and environmental biotechnology, since overproduction of a whole range of chemicals might be improved. However, real application of microbial engineering has to overcome the challenge of setting up a new and appropriate molecular toolkits, since optimized techniques for model organisms might not be fully functional for non-model ones. Recently, new platforms such as CRISPR-Cas have been gaining attention, but it is still not fully developed for bacteria. In this thesis, the challenging way of genetic engineering was taken with the aim to increase biohydrogen production in the sulfovibrio strains. And although several toolboxes seem to be prepared to make easy this scenario, further efforts are needed towards the optimization of a reliable toolbox for genetically modified these bacteria. As for any other scientific contribution, we hope this thesis paved the way for new and more optimal advances in METs. However, there is still some limitations that must be addressed. The first, understanding communication at molecular level between both microorganism electrode and microorganism could gain more insight by using metagenomics and metatranscriptomics. The second, limitation on biofilm growth and biocompatible electrodes might be overcome coupling fundamental and physiological studies with new and promising technologies such as 3D printing. And third, the short product spectrum production rate and selectivity focused on the production of low economical value compounds could be managed with the utilization of synthetic consortia and genetic engineering. But these two applications are still subject to intense research in the points exposed since now, especially in the first one. Now, having all the work presented, I will read the concluding remarks of this thesis. Methanobacterium species were the main biocatalyst in an electromethanogenic reactor. Selected nickel-iron hydrogenases were constitutively expressed. Electroactivity and the ability to increase biotic production of hydrogen seem to be a common trait for anaerobes. The sulfovibrio species were the most interesting strains for a sustained biohydrogen production over time in biocathodes. Improvements are still needed to establish a reliable protocol for genetic modification of the sulfovibrio, and acquired knowledge in this thesis will contribute to new and promising experimental approaches in microbial electrosynthesis. So thank you so much for your kind attention, and now, if you don't mind, I will stop sharing the presentation.
Thank you so much, Elizabeth, for the very clear presentation. And we will then start with the discussion. And starting with discussion is Professor Elaine Vu. Please, Professor Vu, I give you the word to
thank you.
Discuss with the candidates.
Thank you very much, Ellie, for a very nice presentation, very comprehensive, and I must congratulate you. You've achieved quite a lot through this PhD thesis, and because my background is more like from the electrochemical side and also maybe engineering side, although I do quite a lot of work with bacteria, so I actually learned quite a lot from your thesis, especially with the microbiology side. So congratulations for really nice work. So I think for the question wise, I want to ask first like a general, because for your work mostly you're focusing quite a lot on the hydrogen. So we all know this hydrogen is very, very important. So one thing is in this system, one question, because you mentioned that the electron transfer mechanism, there are direct electron transfer, mediated electron transfer, and the indirect electron transfer, which you mentioned, it's hydrogen and the formate. So let's say that the indirect electron transfer, if there is no extra potential like applied, no extra, external like energy source applied, how do you think this process is going to still work similar way?
If I understood you well, are you asking about if I reduce the potential in the systems? So if I use the less reducing potential?
Yeah. So let's say if there's no applied potential. So I know you did this like something like an OCP, right? But the thing is that have you done without it's OCP, but you have hydrogen in the system.
Yeah, but your question is related if we don't have this hydrogen produced biotically in the system, what would happen if we have this situation? Are you asking about that?
Well, I'm asking because you mentioned the electron transfer route, right? Yeah. Mm-hmm. So your indirect mostly with it's through hydrogen, yeah?
Yeah. Mm-hmm.
Yeah. So if let's say you have hydrogen, but without any applied potential.
Oh, okay. Okay. If we don't have the electrons available in the system, what would happen?
Yeah.
But if mainly we have indirect electron transfer and hydrogen is available, the system will work, I think, in optimal conditions without the electrons, because if this is only the main pathway that is working in the systems, it doesn't matter if electrons are present or not. But since it's possible that both conditions, direct and indirect electron transfer, and can be at the same time in the same systems. Maybe with monocultures or pure cultures, we don't see any effect, but with a mixed community, we see an effect because some of the bacteria can use these electrons and some others can use the hydrogen. We didn't test it, but I think that if we could the electron flow and it's still hydrogen in the system, it's just like culturing microorganisms in our conditions in the lab, just in autotrophic conditions.
Yeah, great. Yeah. So basically, without that, so those bacteria, they can still get energy from hydrogen or formate. Yeah. So,
yeah. Sure.
Yeah. Great. Thank you very much. I think another question is about your setup. You have this 3-neck bottles. It's quite interesting. One question, I think what I noticed is you have a very small anode compartment and a very big cathode compartment. So can you just explain a bit how you decide to, those ratios? Because normally for the electrochemical system, you don't want the counter electrode to be the limiting factor. But of course, we know that the cathode side is biotic, so the activity is not that high. So possibly, but how you decide, this, the anode is so small and the cathode.
Yeah, the thing is that this system was built to sustain this bacterial activity in the cathodes, and also it's a work that we did in collaboration with LEQUIA, that they are they have the best knowledge in this sense. And the thing is that we wanted to increase as much as possible the volume in the cathode side, but not affecting this transference of protons between the anode side from the cathode side. And we use the counter electron made of a graphite rod that is a sacrificial anode, and we replace it when it was consumed. Anytime that the root it was consumed, we replace it with another one to have the system still working for some time. But the idea was to maximize the volume in the cathodic chamber and being able to put the 4 electrodes, because there's a lot of space needed for 4 electrodes in the same chamber, and to sustain this methane production in the cathode.
Right. I mean, if I may just ask you, so you mentioned this anode sacrificial, electrode. So what kind of like a reaction are you expecting on the anode side?
Well, in the anode side, we only expected the hydrolysis of the water, so the production of oxygen and protons, and mainly the oxygen could escape with the small gas chamber that we put it. And the protons, we use the cationic exchange membrane to facilitate this transfer between the anode and the cathode.
Okay, so and you expect when you have this oxygen evolution reactions, and then the carbon actually will be oxidized, because you mentioned sacrificial, so it's actually reduced. The road is getting smaller.
Yeah. The road breaks down with used and we replace with another one, a new one, when it's not be able to use the previous one.
Okay, that's very interesting. So if it's oxygen evolution, would it actually be better using a metal, electrode? If the oxygen evolution reaction is the main reaction you expect on the anode side, so that you can actually reduce the overpotential, because if you on the carbon you also break down.
Yeah,
it seems lots of like resistance or lots of things going on. So if you have, let's say, simple like platinum or even say some iridium oxide as some kind of like a oxygen evolution anode, would that actually help to get the whole system like more energy efficient?
Yeah, for sure that if we change the material of the anode, we could reduce this oxygen and also reduce the potential that we used in the systems. But the thing was that oxygen was minimal during all the experiment in both anode and cathode compartments, because we measured sometimes. And the possibility is there to use some of these materials, but in our case and for our objective to use -1 volt of set potential, we decided to use this potential to maintain this availability of hydrogen for microbials to growth and to form this biofilm in the cathode. So it's still a possibility to use other anode materials and see what happens if we reduce the potential and apply the same conditions with a community similar with the one that we have it. And maybe the results or the analysis could result in more attractive ones. I think.
Yeah, great, thank you. What I'm not saying this is So what I just want to mention this is because it seems compared to the cathode side, it's so small, and you have quite complicated process on the anode side. It might be okay because the bacteria is not that active, but it could be so mostly you don't really want to have the counterpart as the limiting factor. So because your anode side actually generates protons, it will be used on the cathode side. So and a better anode, to be sure, you have the ion transfer between two electrodes, and it might actually provide you a better control of the system. So that's all I want to mention. Yeah, great. Thank you very much. And I think the other thing is that with this one, so it's quite nice. You show like for cathode, you show they have a different. I think the abundance of the methanogens is slightly different. So could you just explain a bit why they are different? What caused the difference? These
well, first we have to account that the results are shown in relative abundances, so the grids of sequences for each cathode were a little bit different, but still the differences between the amount of methanobacterium in each cathode resulted in no significant. So it's more about the analysis that we perform, that not that in the reality there's more methanobacterium attached to the electrode in some cases than in the others. So the analysis only revealed that the cathodes were fully colonized by almost 100% for this bacteria. But it's more about how we treat the data, that not the reality in the reactor.
Yeah. Do you think the distance of each cathode between the cathode and the reference electrode could be the factor affecting, causing those small kind of variation?
Probably that's an explanation also, that this distance could affect in the distribution of the microorganisms, or also if some electrode produce more oxygen than another, maybe more microorganisms could attach to it. Yeah, for sure.
Right. Okay. Thank you. I think I have another question. There was one, I think in your chapter 2, there was a CV showing. Yeah. So 56. Sorry. You mentioned this rhodobacteria. So you mentioned the CV. There was a redox peak on this CV. But it seems the oxidation peak we can't really see there. So we only see the reduction part.
Yeah, well, I don't have a clear explanation for that. If you meant with Rhodobacter capsulatus.
Yes.
Yeah. Well, the signal that I show in this graphic in page 56, I think that it was artifactual or something like affected the cyclic, because it's not clear the sigmoid shape of the cyclic. But still, I don't know about the redox potential. Exactly what you ask about the redox potential. Sorry.
Yeah, because you mentioned this is a kind of a redox peak, right? Because it looks like the whole CV is kind of like all in the reduction area. Yeah.
Yeah, I know, but yeah, still this cyclic voltammetry technique is a semi-quantitative one that only explains the activity at some point. So yeah, maybe it's not as good as we expected.
Yeah. So yeah, I'm not really sure, so why. But yeah, so it's only kind of like this only shows the reduction part. They didn't show any. Yeah. So. There's another question. You do the gene. I think in the chapter 3, you do the gene transformation. You use the different method. So you mentioned like those methods you use. Some works well. Some. Are you aware any other like transformation method you could apply?
By now, no, because we take this protocol as the only available one. In fact, this is a protocol that was developed in the lab of Judy Wall, and recently, between the submission of the thesis and today, I contact with the former employers in the lab, and we have the new protocol that it's similar to the one that it's published, and also we have the full composition of the media. So in the last 2 transformations that we did, we used this protocol, and it's the only one described in the literature for the sulfur. So we are a little bit lost right now.
Right. Okay. Have you heard like ultrasound? You can use ultrasound. Maybe also to assess.
I didn't thought about it, but yeah, maybe if we have the techniques here, so we can try it. Thank you.
Okay, great. Thank you. Thank you very much. I don't know how much time I have used, 10, 15 minutes or.
I think you are asking now for almost 20 minutes.
Oh, sorry.
No, that's okay. You can still ask more if you like.
I think, yeah, I just saved some questions for Anya. So thank you very much, Ellie. So I'll just hand over to Anya.
Thank you. Thank you.
Thank you so much, Professor Yu. And then we move to Dr. Annemiek ter Heijnen. Please, Annemiek.
Thank you, Diana. Hello, Elizabeth. Thank you for your very nice presentation. And also, I really enjoyed reading your thesis. So yeah, the topic that you're working on is really, really interesting and important. And of course, it's very close to my heart because I work a lot with electrodes and microbes as well, but a bit from a different perspective as you do. So that also makes, luckily, that I still have some questions left about your work. And yeah, I really want to compliment you with your thesis because it was really nice and easy to read and very, yeah, I think you touched upon a lot of different topics through your thesis, and it's a very interdisciplinary topic, so it's always really hard to, yeah, to approach it from different perspectives. And I really appreciate that you took these different angles, working with mixed cultures, pure cultures, and went through all these processes. It's a real challenge, and it's very nice that you achieved the 2 publications also in your thesis. So yeah, let me raise some questions, and maybe I can start with a bit general one, because in your introduction you mention on page 3 the main limitations of microbial electrosynthesis, and you mention several limitations like the process not being completely selective, the rates and the titers are low. You need a lot of energy input. There are processes, microbial growth processes going on. And the final thing you say there is there's incomplete energy conservation. So my first question is, what do you exactly mean with this last comment on incomplete energy conservation? Can you explain that a bit further to me?
Well, I think that I put it this energy conservation thing at the end of the limitations, more because we are now not able to direct capture the CO2 from the atmosphere and use it for these systems and to close all the cycle. And it was more related in this sense that not that as the production of the energy or the electricity. So maybe the terms are not the best, but I meant to the carbon capture process.
Okay, that's very clear. And I was curious, just out of all these challenges, which one, maybe at the start of your thesis, which one did you think was most important of all those challenges? Because I think there are many. Yeah? So what, in your opinion, was the most important one?
Well, as microbiologist, for me, it was clear that the growth of the bacteria was one of the limitations. To know which microorganisms are able to work in the systems and to exploit these abilities, it's also a challenging thing. So it's more related with microorganisms than with the materials or setups or something like that.
Yes, I can fully understand that. And I'm now curious, because you went through all those steps and all those chapters. So do you at the end still now think that the microbiology is the most important challenge, or did you kind of change your opinion there throughout the thesis?
No, now I think that it's more like a balance between different topics and to couple different technologies, to achieve a better result. So it's necessary the vision of a microbiology, but also the vision of an engineer or a chemistry. So yeah.
Yeah, that is nice to hear. And I agree that this is really a big challenge to combine these different disciplines, and with that, try to move the technology ahead and the understanding of it. So let me continue a bit with the discussion on pure cultures versus mixed cultures, because you raised this very nice topic in the introduction of your thesis, and I think it's also very nice because in your first chapter you work with mixed cultures, undefined mixed cultures, and in the second one you use pure cultures. So can you maybe shortly mention some advantages and disadvantages of pure or mixed cultures?
Well, when using mixed cultures, it is true that the systems are more robust to any effect that could occur in the system. So they are more resilient to several conditions. But it's true that using pure culture, the production is more focused on only one product that could be better or more economically attractive than when we use mixed culture. So from that idea and that advantages and disadvantages to use mixed and pure cultures, we thought that the synthetic culture is the key element here, and we put it few times in the thesis and also during the presentation.
Yeah, and maybe let me pick out a few things that you mentioned, because one thing is you say actually it's kind of easier to control pure cultures because you can also control the selectivity in a way. And if you now look at your results in chapter 1 and chapter 2, do you also think this resulted from your experiments? Do you also see differences there in control of the product spectrum or control of growth between pure and mixed?
Could you repeat that?
Yes, of course, no worries. So you say an advantage of pure cultures could be that it's easier to control the selectivity, for example,
Yeah.
so which product you get. So I was curious, if you look at the methanogens or your hydrogen-producing cultures, do you actually notice a difference in controlling this product spectrum?
No, but by the results that we shown, no, for sure, there's no differences between using the mixed and pure cultures. But it's true that with the methanogens, we only did short time experiments. If we let these times, the community could change and more activities could appear that maybe there were not the best for methane production in this sense. So using the pure cultures, we avoid these interactions not favorable for our objectives.
Yeah, exactly. And I think you also see in your chapter 1 that you have production of hydrogen, but also production of acetate, while eventually most of it is being converted to acetate. Perhaps initially it's also harder to control the selectivity of the reaction with a mixed culture. Yeah, and maybe what I think is also interesting to look a bit at is the stability of the performance of the biocathodes. Can you say something about that related to chapter 1 and 2? So how stable was the performance of your mixed or pure culture experiment?
I think it was more stable in mixed cultures because methane production was stable for all the operation on time. But when testing different strains for hydrogen production, this was completely in the other way. So we had different strains with different behaviors that grew in different rates, and much more complicated to control the specificity. And also I think that to achieve this hydrogen evolution biotically, it was more challenging than not focusing on other compounds such as acetate or methane that could be more easy to produce.
Yeah, maybe we can actually talk a bit more in detail about chapter 2, because I found it a really interesting chapter and I, I have a list of questions. So maybe, so what I found very nice about your approach is that you grow your bacteria and you add already the electrode in the bottle before transferring it to the electrochemical cell. And what I was curious about is, do you see a big difference between the cultures? To what extent in this, let's say, preparation phase or growth phase, they attach to the electrodes? And could you comment a bit on the difference between strains that you may have observed?
Yeah, we analyzed the abundance of these bacteria by qPCR. So mainly purple non-sulfur bacteria were more attached to the electrode during this incubation process than the sulfovibrio strains. And we could see that in one of the plots that is shown in the thesis, where we show the relative abundance of the gene that was attached at the beginning and at the end of the BS operation. So the best biofilm formation cultures were mainly purple non-sulfur bacteria than the sulfovibrio ones, but yeah.
And do you have an idea why? What does that have to do with, or what is the reason for this?
There's a lot of reasons for that. Purple non-sulfur bacteria, in fact, attach to several surfaces, we cultured them in bottles, and the bottles remained green and red for a lot of time. So they are really, really good to perform biofilm. And one possibility is they have this this capacity in the genome to with genes that can be expressed for the formation of this biofilm, and maybe the sulfovibrio strains don't have this type of genes, but I don't know exactly the reason between these differences.
Yeah, I thought it was very interesting that you actually see such a range in differences. And it might be nice to also further explore it later, I think, if you would have the chance. And another interesting, I found it very nice that you did your measurements like after different rounds of CO2 additions, and see how much hydrogen was being produced. I was a bit in doubt myself, so I'd like to discuss with you what is actually the relation between hydrogen formation and the detachment and attachment of the biofilm. Because I think on the one hand, you could say that, well, if you have little biofilm, you could probably have high hydrogen production as well, which would be abiotic. And on the other hand, your biofilm might assist, might help in the hydrogen evolution. So could you comment a bit on how the detachment of microbes do you think influences the hydrogen production rate?
Well, the thing here may be related to the cathode surface itself. So if we had a very thick biofilm, maybe the abiotic production of hydrogen could not be performed in the better conditions. But also it's true that not detecting biofilm, it's not a signal of microorganisms being near to the electrode. So I don't know if I answered your question.
Yeah, I think and maybe related to that, I was also curious. Do suspended cells also play a role in your results, and what kind of role do they play?
Well, maybe it was not the better approach to add the liquid cells, well, the cells in the liquid phase, because we wanted to prove that coated biocathodes could produce hydrogen. But we did it because we wanted to enhance the number of cells at the beginning of the BS operation, and putting both bulk and biofilm cells, we achieved that. And in the pool liquid, microorganisms could do several things that may be not related with hydrogen production, maybe more related with hydrogen consumption. And probably this is one of the explanations that we have with purple non-sulfur bacteria mainly, and also Sporomus aubata.
So that the suspended cells are also taking again part of the hydrogen, right?
Yeah. Mm-hmm.
So do you think that is also the reason that for several strains that in the second round the hydrogen production gets lower? Because I think it was a bit striking that for several strains you saw after the second edition that hydrogen production went down, except for some, which was also very intriguing, I must say.
Yeah, maybe that's the explanation, that the cells in the bowl liquid that also increase between the first and the last sample that we take were doing this activity to uptake the hydrogen.
Do you also have other potential explanations for that? Would it the lower activity or lower hydrogen production in the second round?
Well, maybe it's a matter of something that attached to the electrode that blocked this production, but I don't have any idea of what could cause this. So,
Yeah. So if you would like to keep these biocathodes stable for a long time, so if you think about application of this, I think it could be really nice if you can make biocathodes that produce the hydrogen at a high rate and in a stable way. So what kind of next step would you actually suggest to create a more stable and biofilm that stays on your electrode, or what kind of experiment would you want to do next to get there?
well, first we have to use the best strains to do that. So I will choose the sulfur-vibrio, and then the thing could be use more large electrodes with more surface, and to improve this attachment of the cells in the biocathode, and for sure only using the coated biocathodes for the next experiments. We could try to change the setup of the BES to enhance some important transference between the anode and the cathode and so on. But still, I think that the main limitation is how these bacteria are in contact with the electrode and do the main function in the systems.
Yeah. Okay, that's I hope you will be able, or your group will be able, to further work on this and create more stable biocathodes. And maybe one final question about them is about the biocathodes, where you controlled the potential of the electrodes at 2 different levels. So do you also think that this control, or the choice of potential, or maybe controlling the current rather than the potential, would be interesting? And what would you deem the most feasible method?
Yeah, maybe that are also some points that we can explore in future applications. So use other potentials, or to use other operation parameters for our experiments. Yeah, for sure.
Yeah. Okay, that would be very nice. Maybe, do we still have time, Diana?
Sure, you have.
Sure. Maybe I'd like to ask 1 or 2 more questions about your methane chapter, because I'm also very interested in methanogens. So maybe we can have a bit of closer look at the graph of the performance during the 40 days or so that you operated the system. So I think this is figure 6 in your thesis on page 35.
Yeah.
You have it there, right?
Yeah. Okay.
Yeah.
Yeah.
So it's nice that you actually run this reactor for quite a while, 2 months more or less. And I was a bit puzzled, or well, actually it's kind of normal. We see the current showing quite a lot of fluctuations. But my question to you would be, what do you have an idea what caused those fluctuations? Especially, I think in the first 33 days, there was a lot of. I see that you changed the CO2 feeding, but are there more factors there playing a role?
Are you asking about this time between day 20 and 25 that the current is very low? Or
yeah, maybe overall there's quite some patterns going like this throughout the.
And the tricky thing was that we had all the 4 electrodes connected to one cable, and we pierced the potential stat then in this one cable, and I think it was not stable enough to have a more I don't know, attractive current element, and mainly these are the main limitations that we have with these systems. Yeah, for the CO2, we did several CO2 feedings at the beginning of the experiment to increase the availability of this CO2.
Yeah.
I think that you asked also for that.
Yeah.
And then, I don't know, something happened, maybe other experiments that I have in course or something like that. And we reduced this CO2 feedings, but still more or less stable from day 25 to the end. That is true that we did a few CO2 feedings, but we were closer to the end of the experiment, and we realized that maybe it was not necessary to do it.
Okay, I see. And also I found striking that it seems after your CV scans that the current becomes quite higher and more stable.
Mm-hmm.
Is that true? And do you know why that is happening?
Are you asking about the CVs?
Yeah, so actually the period from day 38 to the end of the experiment, it seems that the current is really much higher there, right?
I don't have any idea for doing that. We choose to do the CV not at the end of the operation.
Yeah.
It's normally done because at the end we needed this 6-hour exposure of electrons. So we did it here, but I don't know how to explain this increase in the current demand because normally we had that reduction of the current demand. So yeah, I don't know how to.
Oh, maybe also something to follow up with. Always more questions arise at the experiments. So thanks a lot, Elizabeth, for the nice discussion. Diana, I'd like to give the floor back to you.
Thank you so much, Annemiek. And I think now I have also some room for questions. But first, I would like also to extend the congratulations, also already mentioned by the other members of the jury. I think, Elizabeth, you did a very good work, and it was really a pleasure to read your thesis. And something that I think you did very well was the reflection on your work, because you will see also with my questions, I had some questions, but then most of the times you actually reflect later on these questions. So I was like, oh, this is a good question. Oh, no, she already actually thought about it and gave her thoughts on that. So I think that was a very good from your thesis. You come up with comments on what you think and what you think could be improved. So that was, I think, a very critical way to write it. And I think that's very good for a PhD level. So you will see that some questions will, you have already touched upon some of the answers, but I will still ask some of them so that I have a little bit of your opinion live and also a bit of discussion on your opinion. But the first question I had was a very basic one that is not related to your research, but or more microbiology background question, and that's about bioenergetics. And it's in your introduction written at one point, that's where you show the equations and the delta G calculations for the methanogenesis and acetogenesis. And there you say that energy conversion, that methanogens conserve more energy by the conversion of hydrogen and CO2 into methane, in contrast to conversion to acetate done by acetogens. So you are saying that methanogens conserve more energy while they are producing methane from hydrogen than acetogens, right?
Yes. Mm-hmm.
So what the first question is, what is for you then energy conservation by microbes?
Well, here we wanted to reflect more the hydrogen consumption or the hydrogen need for each group of microorganisms in contrast with this energy that is conserved during the reaction.
Mm-hmm.
So if you ask for my opinion, I think
not your opinion, but here you have a reaction, right? And both reactions they use for hydrogen, so that's the same. So you are comparing this, and then one has a delta G that is more negative than the other.
Yes.
So you are saying that one, it's true that the energy available in the reaction that has the most negative delta G, it's true, it has more energy available in principle, right? But is that the same as conservation of energy by microbes? So do microbes conserve the totality of this energy? And is it true for all cases that methanogens will all conserve more energy than acetogens? Think about ATP production.
Yeah, okay. Maybe there's not a direct relationship between these two concepts, and here maybe I could have to include some more detail in this sense. So yeah, I think that maybe there's not true that methanogens or all methanogens are able to storage more energy than the acetogens. Yeah.
So you know about cytochrome containing and the non-cytochrome containing methanogens? Do they conserve the same amount of energy, both of them?
No, they're not. So yeah.
But the reaction is the same, right?
Same. Yeah, that's true. That's completely true. Yeah.
So these are two separate things: the energy that the reaction can yield and the energy that the microbes are able to harvest from that reaction.
Yeah.
And it's true that many times methanogens, they can make up to 1.5 ATPs if they have cytochromes from this amount of hydrogen, and the cetogens can go up to 1. So in that case, it's true. But for the ones that have no cytochromes, the story is different.
Yeah, for sure.
They are not so much better than acetogens in principle.
Okay.
Okay, that was just a detail, and because we are now talking about thermodynamics, and also during your discussion with Annemiek, you were touching upon the or discussing this question between pure cultures and mixed cultures, and you are giving your reasons for what is best or what is worse. So one of the ideas I would like you to discuss is this debate on what is better or worse really, should it be focused? We all know that mixed cultures are more robust, for example, so that's an advantage, but are advantages to be taken alone, or should this always be considered in view of the product that you want to produce? Because methane is indeed thermodynamically the best product, let's say, or the more favorable product to be produced when you have a microbial chain. But if you want to produce other products, and maybe you can give me an example, maybe when you have mixed cultures, it's more difficult to control. So is this discussion about being what is better or worse, pure or mixed cultures, decoupled from the type of product that you want to produce, or should that be taken along?
Well, maybe it should be taken alone, not only focusing on the product that we want to produce, and a lot of other things could be considered to choose one kind of approach or another. So the idea here to show mixed cultures, pure cultures, was to have an idea that maybe if we know better the mixed cultures and we could reproduce some of these results in monocultures, we can achieve the synthetic co-cultures. But still the strategy maybe it's not good to only focus on the product spectrum that we have to choose if we want to start working with mixed cultures or pure cultures.
But if I ask you, if you want to produce methane, is it better to use a mixed culture or a pure culture?
I think a mixed culture, because some reactions could be more favored. For example, if we had at some point some contamination with oxygen, if we had some bacteria able to reduce the oxygen on the systems, the methane production is not affected. But if not, the bacteria will die. So yeah.
So could you say that when you have a thermodynamically favorable compound as the final aim for your process, mixed cultures are better?
Yeah, maybe. Maybe.
Okay, something to consider, but I think there might be a good link or a good basis for choice of the type of process, also based on the process on the product you want to produce. Let's then move on, and I would like you to, one of the questions I had, or one of the considerations I had when I was reading your chapter 1, because there you do a lot of work on designing primers, choosing primers for RT-PCR. So my first thing was, why going for all this work and not just trying to do transcriptomics, because you have a very enriched culture. So why not doing that? And then you elaborate on that. So that's one of the cases that I mentioned that you already thought about it. But now looking at both options, which one do you think it would have been easier to do in the lab? And what would transcriptomics, the one you did not do, provide you the chance to maybe discover in addition to what you did with real-time PCR, with quantitative PCR?
Well, in our case, the main limitation to not use metatranscriptomics, apart from the economical part, it was that the extractions that we perform didn't function as well as we expected, and the RNA extracts were having low concentrations of RNA, and there's a limit of concentration of RNA to do metatranscriptomics. So we thought that the better idea to implement was to use this RT-PCR, because it was easy and we have all the elements here in the laboratory. That's also a limitation for us, because if we don't have these elements available, it's more difficult to achieve our objective. It's not impossible, and we tried to explore other applications with other studies that we are conducting right now, with the experience that we have with this methanogenic study. And that was our main limitation, this low concentration of RNA in the extracts. But it's true that maybe with the chance to use other platforms, not only metatranscriptomics, but also to have more enzymes supputatively involved in this process, and to try to analyze them. It is also a thing that we could explore in this sense.
Yeah, about the concentrations, indeed, in the sequencing companies, they ask for quite high RNA concentrations, but we've done before with lower concentrations. Sometimes it works, but of course it's a risk, and
But also not only the concentration, the quality of the RNA is also important, and our quality was very low also. So we are limited in this sense.
Yeah. Okay, I think you discussed that quite well also in your thesis. So let's move to the next chapter, chapter 2. And I would like to look with you to figure 12. And also we will be looking to figure 12 and also the ones where you have the hydrogen monitoring, so the next figures. So you did this experiments by previously growing the biofilm, right? And then you put them in the cathodes and you start measuring hydrogen. And for me, it was very puzzling sometimes that you had these two additions, and then the first addition you have more hydrogen being produced than in the second. And I start wondering if this is also related to some activity that might be in the cells still because they are in the previous medium from the richer medium that you had in the bottles, right? So one thing was, did you, by any chance, did more feeds that are not shown here?
No. We only perform the operation for 5 days. So yeah, maybe this large or higher hydrogen production rate is mainly produced by the photofermentative residual metabolism of this bacteria, or I don't know. That was one option that we had, but we didn't test for further.
Do you know any type of controls you could have done to check this? Because now you have the completely abiotic controls and ones with cells, the pre-grown cells. So what kinds of controls do you include to really test this?
Are you asking about to add some control?
Yes, for example, autoclaved, an autoclaved cloth.
Yeah, maybe that's an option to autoclave the carbon cloth at the end of the operation and measure if the abiotic concentrations of hydrogen still more or less the same. That's a possibility. Also including some controls with bacteria in similar conditions, maybe utilizing serum bottles and CO2 and hydrogen could explain something more, or try to analyze more, for example, more organic acids that we analyze it, but only a few of them, so we could enhance this analysis to know if CO2 is consumed and hydrogen is consumed to form something that we didn't analyze. Well, the CO2 consumption was not tested, so that's also a thing that we could add in further experiments to explain better this behavior with these species.
Okay, and then for sulfogivri pacchesi, then you have higher hydrogen higher amounts of hydrogen being produced after the second CO2 injection.
Yeah.
And there I was also trying to understand. At first I thought, is it because you have more cells growing? But then also when I look back to the graph, figure 12, initial and final concentrations of the organism are basically the same, right? So it doesn't seem that it's like growing.
Yeah.
So what would cause this increase in CO2, in hydrogen production?
Well,
why was it better after the second CO2 addition? What could have been the cause for this?
Yeah, maybe there's an adaptation time for the cells to the new environment because they were cultured in better conditions using acetate as a carbon source. So changing this acetate for CO2 and also the conditions in the BS with the electrons and so on may have an effect with these strains, and they need more time to adapt and to produce this hydrogen. That's a possibility.
But again, a pity there's no follow-up additions to see what would happen. I have here in the site like oxygen marked in the graph. I just wrote it. I was wondering, is there also any effect, or do you think that in the first time that you put the cathode or the cells in the cathode, do you think there might be still some time that the cells need to adapt also to maybe some oxygen that diffuses from the anode or so? Was this taken into consideration? Did you ever see an effect of oxygen diffusion in your cells?
Well, we test for oxygen in the anode and the cathode side with also a microsensor, like the one used for hydrogen measurements. And it's true that oxygen is present in the liquid phase in the anode, but it was lower, I think, I don't remember exactly the numbers, but it was lower than 5 ppms, and in the cathode side, it was lower than 1 ppm of oxygen. And when we added the CO2, we saw that the signal of hydrogen concentrations increased over the time, and then when we started the potential stat, while hydrogen is produced, the oxygen was completely removed to this value of lower than 1 ppm, I think. So our systems are really resilient in this sense, because they don't have this intrusion of the oxygen.
Mm-hmm. Okay. Yeah, still interesting. Another interesting thing is the differences in hydrogen production between the 3 different strains of the sulfur-vibrio that you tested, because they are all sulfur-vibrio and all. Yeah, I don't know variations in the sulfur-vibrio. Of course, they are different species, but I was also then thinking, did you compare the genomes of these 3 organisms, and did you try to find what is different between these 3, or what is Pacesi has that makes it a little bit more special for this?
We didn't do it, and the thing was that for the sulfur-vibrio Pacesi, there's not a complete genome. It's only a draft genome that we can trust partially. But yeah, for sure, we can do this kind of analysis to know which genes are different between the strains, and being more aware of what can be suitable to further study in new projects.
Because in the next chapter, I think you looked a little bit into the hydrogenases, right? That how many hydrogenases these organisms have. And you did not compare that with the other sulfur-vibrio to see if they have different numbers or different types of hydrogenases.
No, we compare only the sulfur-vibrio vulgaris with Pacesi, and focusing only on the hydrogenases and the cytochrome. And the objective was to look if they had the same genes or not. That was the main objective.
Very good. Then jumping to the genetic engineering part, because this was, I think, a big jump, and I wondered if it would have been useful to do an intermediate step between identifying that Pacesi is a very good hydrogen producing strain and going to the genetic engineering approaches. Would there be room also to make a study, for example, again, using transcriptomics, in which you really see which of the enzymes or pathways are very important for hydrogen production, so that you could have a basis for choosing what you want to modify?
Yeah, that was the first idea, to use molecular tools to know more about Pacesi, but time it was the main limitation here. I need to do this 3-month secondment abroad, and we decided to skip this part. That in fact it is necessary to do it, and I missed this part in the thesis, but we skip it, and we focus on reviewing literature to make sure that the gene selection was more or less the more appropriate for applying the genetically modification.
Yeah, and of course you were still busy developing the tools and so on to modify.
Yeah.
So that's always useful work, I would say. Very difficult, I think, and I dare have a bit of a technical question that you also then come up with, and it's the antibiotic resistance. When I was reading your paper or your chapter, it caught my attention that you say, well, there's no information on natural resistance to antibiotic for the archaea, so we took the things from we took the information from other sulfogrivious species. And then I thought, but if this is an important step in the selection of the strains that you want, or on the mutant strains, why not testing antibiotic resistance beforehand? And then you come back to that, and I think now you even tested it because you showed that in your presentation, and maybe that's not the key thing. But would you do it the same way, or now if you do this again, would you test this before?
No, for sure not. If I have the possibility to start again, we will start it with the medium that was described by Geller and co-workers, because this is the suitable medium, and the first step would be the sensitivity test for sure. Yeah.
And then I think you also showed a little bit in your presentation, but in table 23, that you have the summary of the results, the transformation results.
Yeah.
I have just a technical question on the colonies on plates and the growth after picking a colony. Yeah. That means that you is the step, normal step we do, like taking a colony and putting in another plate.
Yeah.
See if it grows.
Yeah, this is.
Because then you have growth on tubes, but normally you do it the other way around. Normally you take a colony to another plate, and then from that to tubes. Yeah. And I was wondering how you do this.
Yeah. The thing here was that after the transformation, we plated the cells, and also we inoculated some liquid. So we did both ways, plates and tubes, and then from both. But here we only shown for the results for the plates. We take from both the plates and the tubes, and we incubated in more plates or a liquid medium as we needed. Yeah.
And then the other question there in that table was for the line test numbers, tests with archaea.
Yeah.
You got positives for growth in colony and also in tubes. Yeah. But then you did not try to see if you could recover the plasmid, I think.
No, that was a mistake in this test C, because we saw growth after picking colonies, but only using the lower concentration of antibiotics. So we discarded the place for that reason. So that check, it's
not a check for all the concentrations. It's only a check for the 25. That's what I understood also from your presentation today.
Yes.
Yeah. Okay. I think I will still ask one more question, and then I will stop, because you already have, I think, enough questions. But when you test Sporomusa, of course, hydrogen then is being converted, so you don't have hydrogen production. But I was then thinking, you also showed in your slides biorecover objective that to have hydrogen producing strain together with an acetogen to produce all the products. So in that objective, is it really better to have this hydrogen plus acetogen, or the hydrogenogenic organism plus the acetogen, and why then not having a Sporomusa that does also the full conversion? So why do you say that's not a good approach, having the acetogen? And why do you want to have really the compartmentalized reaction?
Yeah, the thing here was that we thought that the GMO microorganisms could be able to use the hydrogen produced for the other strain that we selected. But in fact, to move from this idea to use Sporomus albata as an acetogenic microorganism, this is also a good approach, because acetate also could be used for chain elongation by other microorganisms. So yeah, maybe that's a good point and a good approach to explore. And for the compartmentalization part, well, we thought that the two players could be placed in these different positions in the reactor, one in the biofilm and one in the bulk, because it seems easy to control the reactions and so on. But I don't think that's a realistic approach, because with the GMO strains are able to form the biofilm, but also other acetogenic bacteria could form this biofilm, and we would have a mixture between the hydrogen producer and the chain elongator in the biofilm. So maybe it is a way to represent the project, but not a realistic one.
Okay, well, thank you very much. I will not ask more questions. I'm satisfied and had a very nice discussion with you, Elizabeth. Thank you so much. And I think now it's the time to ask the audience if there's any doctor in the audience who would like to pose a question. Please raise your hand. I don't see any hands. So since there's no hands, I will temporarily close this session, because the jury needs to withdraw now for the discussion and decision on the quality of the thesis and the defense. So we will take approximately 10 to 15 minutes and then come back to you with the final result of the discussion. Thank you very much.
Okay, thank you.
So good afternoon again, everyone. The jury has been discussing and talking about the thesis and the defense of Elisabeth Perona Vico and has come to a conclusion. So I'm here to, on behalf of the jury, to convene what that conclusion was. So we unanimously thought that Elisabeth had done a very good job in gathering different types of experiments, taking different approaches. She was very critical in the way she did her experiments and could discuss them in a very constructive way with the elements of the jury. So that was all of very good quality. As a final decision, and considering all these, the thesis, but also the defense today, we have decided to give you the mark of excellence and to grant you with the PhD or to give our allowance, let's say, for the PhD degree. So that's very good news. Congratulations, Elisabeth, also on behalf of everyone. And congratulations also to your supervisors and people who supported you throughout this process, because they are also very important and also very happy for you at this moment. I would like to ask Elisabeth if you want to say some words.
Yeah, thank you so much, Diana, Annemiek, and Eileen, for being here today and make this defense more easy for me. And now it's the time when I'm supposed to say something meaningful, and I wanted to do it in Catalan if you don't mind. So, yeah, moltes gràcies a tots per estar aquí. Gràcies als meus pares i a l'Albert, perquè són els que més m'han aguantat, als meus directors i a tots els que m'heu acompanyat durant aquesta aventura. I res, dir-vos que tindrem una mica de pica-pica aquí, que podeu dinar i que podem compartir una mica més de temps junts. Que moltes gràcies.
Elisabeth, I hope you can enjoy the rest of the day with your close colleagues and with your family. Congratulations again. Maybe also Annemiek and Elaine want to say congratulations to you. Yeah, from my side, I just wish you the best for your future career.
Thank you so much.
Yes, congratulations, Elisabeth, and hope to meet you in person in the future.
Congratulations, Elisabeth. Elisabeth, very well done. It's a very, PhD is not an easy journey, but you've done that. Hope you enjoy now. So Dr. Perona Vico? We may. Okay.
Okay.
Have a good day.
Thank you. Thank you.
So thank you so much also to the elements of the jury. Hope we can one day meet together in person and still celebrate a little bit, Elisabeth. Have a good day, everyone, and I think we close the meeting.
Yeah, thank you very much, Diana, Elena, Annemiek. It has been absolutely pleasure to have you on board and to make the things so easy. And of course, to congratulate Elisabeth for this really outstanding presentation. So with that, we close the floor. Thank you very much.
Thank you. Bye.